صباح الخير. Okay, good. طيب, within a few seconds to start. Just بس غريب انه انا مش سامعكم, مش عارف معمول اوتو ميوت يعني للكل. I got this. السلايد طالع عندكم يا جماعة؟
طالع الدكتور.
تمام, يسلموا, شكرا. يا ريت حدا يتفاعل معي مشان ما احسش انه لحالي, لانه ما فيش صدى صوت. اوكي, ثواني بس خليني اسكر الباب. يعطيكم العافية مرة ثانية. نكمل الموضوع اللي كنا بدأنا فيه المرة الماضية, اللي بيحكي عن digestion process from the biochemical point of view. كنا حكينا انه في عندنا digestion of different molecules, so that's why we are classified or actually categorized the chapter or this lecture to different. So we started with the carbohydrate story, mentioned that we have different types of transporters of the digested carbohydrate molecule, and the abundant one as usual, the glucose molecule, and we have such kind of GLUT transporters, and we have like more than 14 of these GLUTs, because we have specificity or tissue specific of these. As an example, you can see here GLUT number 5 is abundant in intestinal epithelium cells as well as spermatozoal cells due to the lactose for such kind or to be transported in these cells. Okay, so how the GLUT transporters actually expressed on the cell, we have to remember that these molecules are already synthesized in these cells and stored in a vacuole structure, and they have to be expressed on the surface of the cell according to certain signaling molecules or signaling mechanism, if by insulin, as we can see here. So the insulin, if we have GLUT responding receptors, so there is a kind of pathway or signaling pathway that will stimulate the expression of these molecules that are already synthesized in the cell on the surface, and it's not going to stimulate the synthesis of the GLUT molecules. It's just a process of the stimulation of the expression of these molecules on the surface of the cell, glucose absorption to these kinds of cells. Just to compare the two different types of tissues that we could have in our body, in order to discuss the need of expression of certain GLUTs or not. So as you can see here, we can see the difference between the neural cells or neural tissue structure compared to the non-neural cells. We can see the presence of these gaps. Basal membrane here, spatial gaps that can allow the transportation of glucose or other solutes actually. But if we go through the neural cells, we will see what's called blood-brain barriers or blood barriers that will prevent the transportation of a lot of molecules or a lot of solutes to the neural cells or neural tissue in order to avoid the toxicity. So there is a special mechanism to transport such molecules. That's why we need much more complicated if you follow these keywords, if you would, or key points, you can see there is a tight junction, most abundant or more abundant in the neuron tissues, as well as at number two, you can see the narrow intracellular spaces compared to the non-neural tissues, and lack of pinocytosis process, which is available in other types of tissues, and continuous basement membrane. As we mentioned before, there is no spatial gaps here, but we have them in other types of tissues, and the glucose transporters are found on both basement membranes. This is due to the sensitivity of such kind of tissues. Moving to another kind of molecule that needs to be digested by our digestive system, and we will focus on the biochemical mechanisms that are responsible for the digestion of these molecules, and it will be the lipid molecules. So there is a process of digestion and absorption of these lipids, and the most abundant molecule, the most common molecule that we could have as a lipid molecule or fat will be the triglyceride. And you are familiar with this molecule. Basically, it consists of the glycerol molecule, which is not a lipid, by the way, it's alcohol molecule that is connected to three fatty acids through these ester bonds, as you can see. So this is an ester bond as well as the other one and the third one. If you have what's called tri that are connected to glycerol, of course, you will have what's called a triacylglycerol or triglyceride molecule. Okay, so this is the major fat component that we could find in the human diet. So it will be the most discussed one, and by the way, it's the simplest one to be digested as a fat molecule. The a new thing that could be introduced to some of you uh, is the من أيام المدرسة they said that the digestion of lipids starts in the small intestine, and what they meant by that is most of the lipids or most of the digestion process is going to be conducted in the small intestine because we will have the lipases that are responsible for the digestion of the lipid. However, we have, for certain extent, of course, digestion process in the mouth that will start in the mouth as well as the stomach. So as I mentioned before, it's for certain extent. It's not the major one. So we could have what's called mouth or oral digestion of lipid, and it will be gastric one in the stomach, because we have these two enzymes. One of them is called lingual lipase, which is going to digest short and medium chain fatty acids that are in the triglyceride molecule. Don't forget, we are going to cleave these fatty acids by these lipases. So this is the point. We want to cleave these bonds or the ester bonds. So we have lingual lipase, which is secreted by sublingual glands that are located in the bottom of our tongue. And these lipases can digest a certain amount of triglyceride molecules, but not most of them, of course. And we do not rely on these enzymes, but they are there, as well as the gastric lipase enzyme or acid resistant lipase or acid lipase, which can also digest short and medium fatty acids that will be found in triglyceride molecules. Why we need such kind of odd mechanism or odd digestion process of lipids in the mouth or in the gastric cavity? This is because we have at certain period of our developmental life, we will rely on the milk of our maternal origin. يعني معنى آخر لما بنولد إحنا بنكون لسه نيو بورنز، معظم غذاء راح يكون عبارة عن حليب لغاية فترة مش قليلة، وبالتالي بتطلب هذا الموضوع إنه process of digestion of the fats that we have in the milk as well as the other components like the proteins. So that's why we have such kind of these enzymes that will help the newborns or the infants in the digestion process of lipids, as well as the acidity of the stomach for the newborns is less than the adult one. It's lower than the adult one. I mean, the acidity is not that much as we have in adult stomach. بمعنى آخر إنه الحامضية تبعتها أقل شوي من الادلت, so it will help in the digestion of the lipid at that site. Later on, when we develop as an adult individual, we will have or we will rely on other types of lipid digestion mechanisms that depend in a complicated mechanism, which is also available in newborns, but it will be the major one or going to be the major one in the adult, which rely on the lipase enzyme, and this lipase is going to be secreted by the pancreatic cells with other digestive juice components. The strange thing, or the actually different thing of the lipid digestion compared to other molecules اللي هم البروتينز والcarbohydrates or even the nucleic acids, is due to the difference in the solubility of these molecules. As you know, in lipids are hydrophobic molecules compared to the other molecules اللي هم البروتينز والcarbohydrates والnucleic acids. هلا هذول المولكيولز الثلاثه عنهم مؤخرا هم عباره عن water soluble molecules, which means that انه الانزيمز اللي هم اصلا water soluble molecules as well, they can access or they have access to reach the bonds between the building units اللي هم monomers in order to break down the bonds, if it's a tight bond or if it's a glycosidic bond or phosphodiester bond in the case of the nucleic acids. While here we have a barrier of hydrophobic shell, if you would, or hydrophobic structure of lipids that will actually an obstacle to digest these lipids. So that's why we need another process of helping mechanism for the digestion of lipid, which is the bile salt or bile acid molecules. ليش بنسميهم salt بنسميهم acid? It's the same molecule. When I say bile salt, it means that it's conjugated with sodium, for example, as precipitate. It can be a salt. And when it's a solution or solutory, it will be an acidic form. So that's why it's called bile salt. Okay, so this is an example of the bile salt or bile acid at the same time that could be found or secreted by the gallbladder, and primarily it was synthesized in the liver. So the gallbladder is not responsible for the synthesis of the bile acid or bile salt. It's responsible for the secretion, actually starting with the storage process. So it will store the bile salt and then it will secrete it to the digestive cavity due to certain signaling molecules or signaling hormones that are responsible for the stimulation of the secretion or release of these components to the duodenum area, as well as there will be enzymes that are going to be secreted from the pancreatic juice components in order to digest other molecules besides the lipid one. So basically these bile salts اللي هو بحالتنا هي اسمه الكوليت or cholic acid is responsible for the emulsification of the lipids in order to facilitate the digestion process by the formation of what's called micelles. So the micelles عبارة عن if you would globular structure. It's not just ball structure. It's just globular. It means that it can have irregular structure, but still globular. It means that it's going to be soluble in the water. And there will be these components that you have, like the lipase enzyme that is secreted from the pancreas with colipase. So these components are going to be secreted together because the colipase is responsible for the activation of the lipase in order to digest the triglycerides or triacylglycerol molecules. Other lipases that can be found also in the gastric juice digestive components اللي هم phospholipases, which is responsible for the digestion of phospholipids, as well as cholesterol esterase, which is responsible for the digestion of the cholesterol ester, which means that if you have cholesterol with fatty acid that is connecting the cholesterol at this site, this enzyme is responsible for the digestion of this
So briefly, what's going to happen is the following. You will have this component of the diet, which includes the triglyceride, basically. And we will have here the bile salt as well as the digestive enzymes. And eventually, you will have free fatty acids after the digestion, as well as a molecule of what's called monoacylglycerol or 2-monoacylglycerol. Okay. So what we are going to do as a digestive system is the following: digestion or breakdown of this phosphodiester bond, and you will get, or the third one, okay? And eventually you will break either carbon number 1 or carbon number 2 esters. So you will have this final structure, which is called monoacylglycerol, and the lipase enzyme has no access to reach the second carbon fatty acid. So what you will gain from the digestion of the triglyceride is 2 free fatty acids and 1 monoacylglycerol molecule or 2 monoacylglycerol molecule، لانه على كاربون نمبر 2. يعني مش مهم الاسم انك تسميه monoacylglycerol او 2 monoacylglycerol because by default it means the same molecule. So after that, these components are going to be absorbed by the endothelial cells اللي هم lining the ileum cavity or the gut system. And after that, these endothelial cells are responsible for the resynthesis or reformation of the other types of lipid complex structure in order to be absorbed by the lymphatic system. And then after the lymphatic system, it will reach the blood. So the critical points here is is the absorption of the fatty acids. It's not going to be through the blood system, lymphatic system, and then to the blood system. And the reformation of large molecule of lipids, which is called nascent chylomicrons. Okay, so to make the story short, these endothelial cells are responsible for the rejoining of what we digested at the digestive system to form which contains the triglyceride molecules, phospholipids, as well as cholesterol ester, and conjugation of certain types of apolipoproteins. Why we are doing that? In order to increase the solubility of these lipids, because as you know, lipids عبارة عن hydrophobic molecules, so it will be not easy to be dissolved in aqueous solution. That's why we need to conjugate them with other types of polar molecules اللي هم البروتينز. And once we are doing that, we are molecule easy to go through the membranes, so it will not be diffused. And this is very important concept. There is no diffusion process. It will be okay. I توقعت انه في سلايد ثاني كنت انا يمكن بالبيسك نشرحه اكثر. So because this molecule is large, so it needs a process of exocytosis rather than diffusion by these endothelial cells to the lymphatic system. And then after that, these chylomicrons will be transported to the circulatory system or the blood system at the end of the day. Okay, so basically we have these molecules that are going to be digested. If you have cholesterol ester, you will have cholesterol esterase enzyme in order to release the fatty acids from the cholesterol. And here we will have phospholipases, and we have different types of phospholipases, phospholipase A2 or C or T, that are responsible for the breakdown of the other components of the phospholipid molecules. And eventually you will have phosphatidic acid as well as free fatty acids. And this molecule in this case is called lysophospholipid. Okay, if you digest it by just one enzyme, which is called phospholipase A2. Something very interesting when we talk about the bile salt, and we have to mention that because it's important. The bile salt itself can be recycled from the, and we will have like 95% as a secondary bile salt. What does that mean? Secondary bile salt, it means that the bile salt that you already secreted to the digestive system, and this one is called the primary bile salt, and this amount of bile salt is called secondary one because it's going to be reabsorbed. At the same time, we will have like 5% of the bile salt that is going to be ceases. The important thing is the bile salt, it's by itself is inhibitor to the lipase enzyme. هلا واحد راح يقول لي طيب البايل سولت احنا احتاجناه عشان نعمل emulsification للليبيدز، بمعنى آخر نزيد الذائبية تبعت الليبيدز عشان نعملها digestion أسهل. كيف ممكن يعمل هو inhibition إذا هو بيعمل facilitation or facilitating process to the solubility. هلا هذول موضوعين مختلفات تماما. هلا بيسهل الذائبية at the same time نفس البايل salt is not facilitating lipase. That's why we have the colipase protein, okay, as an activator molecule to the lipase. So this is the mission of the colipase molecule, because we have inhibitor like the bile salt, which has been found to be inhibitor for the lipase enzyme. Going to or moving to another molecule that is going to be digested and absorbed by our digestive system. And to make it simple, we have protein is the stomach. Until today, هذا اللي بنعرفه إنه البيبسين is the starting enzyme for the digestion of proteins, and it will generate peptides, small peptides or oligopeptides. And as you know, the pepsin as well as the HCl are secreted from the gastric cells in order to start the digestion of proteins. And the HCl itself is activating the pepsinogene form. So basically, what we have is inactive forms of these enzymes. They need to be activated in the site of the function. So the pepsinogene is secreted as inactive enzyme, which is called zymogene or proenzyme. This is the inactive form of the enzyme, and it will be activated by the acidic condition in the stomach. Is also there is kind of misunderstanding here. The pepsin is going to activate the conversion of pepsinogene, not the pepsinogene itself. So this arrow should be in this side, but it means that or implies that the pepsin can do what's called self-activation. هلا البيبسين مختلف عن الاشياء الاخرى انه they are secreted from the pancreatic cells. The pepsin is secreted from the stomach cells, and here we have these kinds of enzymes that are secreted from the pancreas, or the will have from the pancreas is the trypsinogene, and it will be activated to trypsin, chemotrypsinogene, to form what's called trypsinogene. And the trypsin, as you can see here, is the major activator of other enzymes or other zymogene enzymes, except the trypsinogene itself. It's activated by enteropeptidase enzyme, which is a component of the digestive enzymes that we have in the digestive lumen. The proelastase enzyme is another enzyme secreted from the pancreas, and procarboxypeptidase A and B, which are responsible for the digestion of certain specific sites of the proteins. So all of these enzymes, by the way, when they are activated, they have what's called specific peptide a random way. Okay, because and this is somehow different from the digestion of carbohydrates. هلا بالcarbohydrates المajority راح تكون عبارة عن polyglucose molecules. So the bond is very similar. It's the same bond اللي هو alpha 1 4 glycosidic bond. So you need just one type of enzyme for that kind of bond, unless you have branches, of course. هلا بالproteins the story is completely different, because we have different types of amino acids that are forming these peptides to match the specificity or match the bond between two different amino acids. For example, إذا كان عندك ألانين وفالين جنب بعضهم بتختلف هاي الرابطة لو كان عندنا ألانين وجلوتاميك أسيد. Okay, so it needs another kind of enzyme. That's why we have different enzymes for the protein digestion, as well as at the end of the day we have what's called aminopeptidases, as well as di and tripeptidases, in order to secrete or absorb single amino acids to the blood system or to the circulatory system. Okay, so when we talk about absorption, you have to remember that we only absorb monomers, not dimers, because dimers actually or larger molecules are antigenic, and they will cause certain kind of sensitivity in the body, because they have antigenic properties that will stimulate the immune system. That's why we have to complete the digestion process until we reach single residues, whether amino acids or lipid molecules إذا جاز التعبير بالنسبة لللipid molecules or how we activate our enzymes. Again, we have Schiff cells that are responsible for the secretion of the pepsinogen that will be activated to pepsine by the acidic condition in the stomach. And the acidic condition is generated by the secretion of hydrogen and chloride, not the secretion of HCl itself من الباريتال cells. So parietal cells ما بتفرز HCl. It secretes or they secrete hydrogen and chloride, and when they are found in the solution, that will be responsible for the activation of pepsinogen to pepsin, as well as digestion of the protein by the acidic condition itself. Pepsin has what's called a broad specificity rather than specific sites or specific locations of amino acids. And in order to understand the specificity story, we have to take a look to this peptide, for example. ومحدش يسألني بعدين يبعث لي بالإيميل إنه هدول specific sites حفظ ولا لا. At this for example, the aminoperoxidase is responsible for the digestion of any peptide bond that is followed by phenylalanine, tyrosine, glutamic acid, or aspartic acid. Okay, so it breaks this kind of bond at the amino site when we have this amino end. Pepsin is breaking such kind of bond when we have these amino acids in the left side, I mean at the amino side of the peptide bond, while the chymotrypsin is responsible for the digestion of the peptide bond when we have this group of amino acids at the amino site. When I say amino site يعني من جهة الان جروب، okay؟ مش من جهة الكاربوكسيل جروب. هلا إذا بنيجي على الكاربوكسي بيبتيديز، وهذا السبب إنه في عندنا امينو كاربوكسي بيبتيديز. الكاربوكسيز is responsible for the digestion of the carboxyl end اللي هو C end when we have such kind of بيبتايد. فبالتالي في شيء عندنا خاص للامينو تيرمينال وفي شيء خاص للكاربوكسي تيرمينال. That's why they are called aminopeptidase or carboxy peptidase enzymes. بس تعرفوا انه السبيسيفيسيتي is not abroad and it's not that specific. ايش يعني that specific؟ يعني مش خاص بنوع واحد من الاحماض الامينيه. You could have group of amino acids that have similar properties in order to be digested by these proteases. Something very interesting about the digestion of proteins as well as the carbohydrates, as we have seen before, إن في عندنا enzymes موجودين على the cells, and they one amino acid at the time, aminopeptidase, as well as the carboxy peptidase. The intracellular peptidase, in order to absorb single amino acid to the circulatory system. هلا نحكي شوي عن بعض القصص اللي فيها clinical correlation, and they are very common, very simple to remember or to recall when we start to talk about the digestion of carbohydrates. So there are some individuals with genetic deficiencies of the saccharase isomaltase complex اللي بتذكر الستوري هاي معنا انه عباره عن كومبلكس، وهدول الناس اللي ما بيقدروا يهضموا السكروز والمالتوز. Actually, the sucrose is the major one. They will show a symptom of what's called sucrose intolerance. What does that mean? They are not be able to digest sucrose, but they will not have a problem with the digestion of other lipids like the starch in their meals. So molecules, so they can digest maltase unless there is another genetic disorder that will be found in such individuals. Ages, they will develop what's called sucrose intolerance. وعشان هيك بصير في عندنا عمليات أحيانا تشبه lactose intolerance in certain individuals. So just remember that we have other types of carbohydrate intolerance similar to the lactose one. And basically they are genetic related. There is a genetic predisposition. معنى آخر إن في عندنا mutations بهذول الانزيمز. Another clinical correlation that you have to kidney, they can reabsorb glucose into the blood in order to have glucose-free urine. And these transporters actually sodium dependent transporters because we are secreting high sodium concentration in the urine. Okay, so they are able to transport glucose against the concentration gradient because they have these sodium glucose transporters. The other cells, compared to the kidney cells, they have passive diffusion, which means that they rely on the concentration gradient. Talking about erythrocytes, and just to mention the story rather than remembering the numbers. انا ما بهمني الارقام الموجوده هون بالنسبه للكي امز الموجوده على الانزيم الترانسبورترز هذول. هلا الكي ام كلنا بنعرف انه عباره عن ميشيلمنتن كونستانت that is related to the enzyme activity، بس هو ما بنطبقه على الانزيمز فقط، بنطبقه على كل البروتينات اللي بتعمل function in fact. So the most abundant glucose transporter that we could have in the red blood cells is GLUT1, and it's insulin independent, and it has a very strange Km value, which is from 1 to 7, which means that there is a broad range of Km, and this is unusual. When you have Km, it will be constant one because it's a constant, okay? Which means that we have a special kind of cell that can absorb actually or transport the glucose to the cells in order to keep the survival of these cells. So as a blood glucose level fall for low concentration, still the red blood cells can absorb the glucose as much as the glucose at high concentration. Here we have another kind of clinical condition that describes Al Martini individual, and he's an alcohol abuse guy, and at the same time he's not eating very well, and at certain time he went to the emergency department, and he has some kind of diarrhea, some pains, and this is after, of course, physical examination. You have to read this because it's very interesting. And at the end of the day, they discovered that Al-Martini has what's called a pancreatic amylase enzyme at high concentration, which means that, or implies that the pancreas is affected by what's called pancreatitis, or there is kind of inflammation in the pancreas tissue that is responsible for the secretion of these enzymes in the blood. So they tested the amylase or lipase in the blood, which implies that, or which will reveal the presence of problem in the pancreas due to the alcohol abuse in this guy. Okay. After a while, actually, this is an answer for this question. When he was finally able to tolerate a full diet, it means that he went back to normal diet and stop alcohol abuse. The answer will be the stool will be go back to the normal staturia due to the inability to digest lipids, because that guy had amylase problem because of the pancreatitis. Future وليس بالضرورة أن يكون هو alcohol abuse. It could be any other problem with the pancreas. حكيت كلمة pancreatitis. Okay. هلا هاي الستوري حكينا عنها اللي هو البايل سولت inhibits the pancreatic lipase. That's why we need colipase in order to activate the lipase enzyme. فهذول البوكسز يعني بسوى انه تطلعوا عليهم لانه بيحكوا عن قصص احنا كنا حكينا عنها basic science, and they are very easy to be read. Okay. Story when we talk about alpha 1 antitrypsin deficiency. هلا هاي القصة حكيناها بالبيسك بايوكيمستري، and again أنا بدي أمر عليهم مرور سريع. إنه في عندنا كونديشن اسمها alpha 1 antitrypsin. الالفا 1 antitrypsin is responsible for the deactivation of elastase. So إذا كان في عندنا deficiency بالالفا 1 antitrypsin, we will have a problem in the facing of elastase. فإذا صار في عندي inflammation باللنج على سبيل المثال، هاي اللي بتسبب emphysema case, and you have high concentration of inflammatory response by our white blood cells, وبلشوا هدول white blood cells يفرزوا elastase. If you are normal individual with normal alpha 1 antitrypsin, there will be no problem or our alveolar cells will be safe. Otherwise إذا كان في عندنا deficiency بالalpha 1 antitrypsin due to genetic disorder, there will be a problem of emphysema due to the action of the elastase that is secreted from our cells on our alveolar cells. فكأننا بنعمل destruction to our cells by our enzymes due to blind deficiency اللي هو الalpha 1 antitrypsin. Okay, and it's the same thing. This is very interesting وبوصلني كثير أسئلة على هذا الموضوع إنه can we increase our muscles by eating amino acids زي اللي بيعملوه بالجيم. Is it the case يعني مجرد بس أكل الأمينو acids as a supplement? No. In fact, these amino acids are just residues that can be converted to other molecules. They can be converted to carbohydrate molecules. And if you convert it to carbohydrate molecules, it means that carbohydrate molecules can be converted to lipids. فالamino acids doesn't mean that you are building more protein unless you are practicing certain exercises. Okay. فالاكسس protein doesn't mean that you are going to make much muscle. You need to exercise these muscles or to build up the structure of the muscles rather than eating the proteins alone. Okay, so this is a very important issue. This is at the molecular level how we activate enzymes from the zymogen form or inactive enzyme to the active one. It's just cleavage of certain piece of the peptide in order to expose the active site. وبالتالي الفرق بين الانactive والاكتف انه بكون المكان تبع الاكتف سايت مغطى, and when we have like acidic condition it will be cleaved to have the active one. This is just illustration of the how we activate enzymes. هلا الهارتنب disease is kind of a disease that was called after the family name. في عندنا عائلة صار اسمهم عندهم هارتنب disease او اسمهم هارتنب. كان في عندهم مشكلة absorption. Have what's called a neutral amino acid urea. هلا من الاسم you can conclude the condition itself. لما بقول amino acid urea, it means that you have amino acids in the urine, and these amino acids are neutral amino acids. Why we are saying neutral amino acids rather than saying specific amino acids? Because the transporters in the kidney or renal system working as general transporters. I mean, one transporter for a group of amino acids. So there is a transporter for neutral amino acids. And if this transporter is defected, you will have amino acid urea or a neutral amino acid urea. It means that these amino acids are not going to be absorbed by the renal system, and they will be released in the urine. And due to the loss of these amino acids, the urine itself is normally not containing glucose or amino acids or proteins or lipids. هذول الماكرومولكيولز كلهم ما بينزلوا باليورين الا في ظروف معينه زي الدايابيتس فور اكزامبل. يو ويل هاف جلوكوز لانه بصير الثريشولد تبع الكدنه منخفض بالنسبه للكونسنتريشن تبع الجلوكوز. هلا الاذر جينيتيك كونديشنز اللي هي مرتبطه بالديفيشنسي اوف ذا ترانسبورترز will cause, for example, amino acid urea, which means that the amino acids are going to be secreted in the urine, which will cause deficiency of these amino acids. وممكن يكونوا هذول essential amino acids ضروريين ل building up of certain proteins, and they could be associated with certain protein deficiency symptoms like pellagra اللي هو عدم القدرة على العمل أو الخمول بسبب هذا النوع من الكونديشن. So it's a very interesting topic, and if you go back to the date of this publication, it's a very old one. It means that we have a history with this kind of deficiency. Okay, هيك بنكون خلصنا الجزء الأول أو المحاضرة الأولى يفترض من التوبيك تبعنا. ننتقل الآن ل second lecture or second part, which is describing. خليني أعمل share هون. Okay, so the second part is going to talk about or to describe the liver metabolism, because the next one or next topic will talk about the liver disorders or liver function and liver function tests if we have problem in the liver. هلا احنا موضوعنا بتكلم عن digestive system itself as anatomical structure. It contains certain essential parts. بس الليفر هو essential but it's not the major one or part of the digestive system itself per se. It's an accessory system that is helping the digestive system in order to complete the digestive process. That's why we are interested in the liver, and we are going to describe certain functions of the liver that are related to the digestion process. هلا بعض الاشياء انا بعتقد انها مكرره لانه موجوده في الاناتومي، انه how many loops or lobules that we have and how much is the blood supply that we have in the liver and the types of the liver cells that we could have. I think you are familiar with these things. وللأمانة أنا ما راح أركز على هذول في أسئلتي, just to أنا حطيتهم هون in order to refresh your memory. You can go through them. And the most important thing is this thing اللي هو major functions of the liver itself. So basically we have a lot of functions, and let's explore them or going through them one by one. The liver is central receiving and recycling center for the body, because the portal system is responsible for transportation of all entrance molecules to the body when we absorb them through the digestive system. And this is because it's responsible for the inactivation and detoxification of what's called xenobiotic compounds and metabolites. So we have a huge number of molecules, and you cannot imagine that how many molecules that we introduce to our body that need to be detoxified or to be inactivated in order to avoid certain drastic conditions that could be developed in our body. So it's very important to be detoxified or inactivated, and this process is going to be performed by the cytochrome system that we have in our liver. And if you go through this story, here cytochromes, it's amazing mechanism. يعني من الاشياء اللي تدلك فعلا على الاعجاز في انه احنا already designed هذا المكان. يعني هاي القصه حكيتها في اكثر من مكان. Already designed as a cytochrome system in our liver or in our cells in the liver, designed for non-formed molecules, not yet formed molecules or compounds. What does that mean? احنا بسبب التطور تبع التكنولوجي والبايوتكنولوجي والكيميكال تكنولوجي، كل يوم في عندنا مركبات جديده، وابسط مثال عليها اللي هو الدراجز، عشان هيك بسموهم زينوبايوتكس، الدراجز تابعين لهي المجموعه. هلا هذول المولكيولز او الكومباوندز اللي بنصنعهم احنا بطرق كيميائيه، they can be detoxified and recognized by our cytochrome system. هلا as a miracle thing انه كيف احنا مصممين حسب التاريخ تبعنا من اذا كان ملايين السنين ولا الاف السنين، designed in order to face unknown molecules or unknown compounds. فهذا الدليل القطعي على قصة الإيفوليوشن، إنه أنت الإيفوليوشن كنظرية، وفي ناس بيقول لك إنه حقيقة وهذا إشي مضحك، كنظرية إنه الإيفوليوشن بسبب process of exposure or process of neglecting something. على هذا الأساس قامت نظرية التطور في إنه في عندنا عملية استخدام لأشياء أو عملية تعرض لمحفزات حيوية، إنه إحنا نعمل أشياء معينة في أجسامنا أو الكائنات تعملها بطريقة لا إرادية أو إرادية. I don't know شو معنى إرادية لما بقول إرادية. ونعمل عملية ميتابوليزم لمولكيولز، بينما هذول المولكيولز أصلا مش موجودين. ف it cannot be explained by evolution، إنه إحنا تطورنا لشيء لسه مش موجود أصلا. Keep in mind إنه in the future we will develop other kinds of molecules and toxic compounds that will be metabolized by our cytochrome systems. فالسايتوكرومز زي ما حكى لي أحد البروفيسورية في وقت من الأوقات is the guide to the God، دليل على وجود الرب، لأنه هذول أشياء موجودين بالأساس لمواكبة أو للتعامل مع أشياء أصلا ما وجدت بالطبيعة. Okay, anyways, The liver is responsible for the regulation of blood glucose level اللي هو بين قوسين homeostasis اللي المحافظة على تركيز الجلوكوز بشكل بسيط جدا هو عملية الجلوكونيجينيسيس تصنيع الجلوكوز أو الجلايكوجينولايسيس اللي هو تحليل الجلايكوجين أو تكوين الجلايكوجين اللي هو glycogen synthesis. ففي عندنا different mechanisms that can regulate the glucose level in the blood. Synthesis of lipids like cholesterol and triglyceride is also another major function of the liver because it's responsible for the formation of what's called VLDL, very low density lipoprotein. To make the story short, كل الليبيدز اللي احنا بنمتصهم من الغذاء نعملهم عملية دايجستشن وبضل في عندنا بقايا هذول البقايا بالسيركوليشن بروحوا باتجاه الليفر itself. The cycling of these molecules and exporting them as a new structure which is called VLDL. فالليفر مسؤول عن تصنيع الليبيدز مرة أخرى وتصديرها إلى السيركولاري سيستم. Ammonia detoxification and urea cycle. Okay, it means that we are going to convert the ammonia or ammonia molecules to urea in order to be less toxic. Ketone body production because ketones are very important for certain kinds of tissues during starvation like the central nervous system. Nucleotide biosynthesis, which is very crucial thing that we have to build in our body. Nucleotides اللي هم الادينوسين guanine cytosine هذول مع الريبوز والphosphate group they are called nucleotides. So this synthesis of nucleotides is assigned to the liver. Synthesis of some proteins that are found in our circulatory system واحد منهم الألفا-1 antitrypsin by the way. Synthesis of glycoproteins and proteoglycans that can be used by other types of tissues and it's a major site for the pentose phosphate pathway اللي هو مسؤول عن metabolism of glucose to produce NADH or NADPH molecules as reducing agents for the anabolism. So these are the major functions that can be conducted by the liver. هلا بعض الفانكشنز هذول مش مع خلايا أخرى. Okay, but the liver is very specific site for the synthesis of these or for these kinds of functions. هلا the liver itself per se consumes like 20% of the body oxygen, which is a huge amount of oxygen, which means that the liver is doing a lot of functions. Okay, and these are the molecules that can be generated in the liver or other types of the cells as energy currency. What we mean by energy currency, we can use these molecules in order to catalyze certain reactions because they are energy currency molecules. However, NADPH is not energy currency, but it's synthesized by the liver as well as acyl-CoA thioesters. هذول molecules helping in the anabolism. Okay. هلا هذول عباره عن energy molecules currency ولكن هذول عباره عن reducing agents المقصود انه هذول كلهم مسؤولين عن الانابوليزم in the بحب اختصر الاشياء انا in order to get the most important point when we talk about any subject. هلا الليفر امام حالتين بالنسبه للميتابوليزم. الحاله الاولى بنسميها well fed state اللي هو في عندنا nutrients and the other one is called the starvation state. So if you clarify these processes according to what I am saying here, it will be much more easier to understand the function of the liver itself. هلا بالfed state or well fed state or diet state and the nutrients, the blood is going to supply the liver with those molecules. Glucose اللي هو جاي من الكربوهيدرات الامينو اسيدز من البروتينز والكايلومايكرونز اللي جاي من lipids after the digestion by our digestive system, our cells, we will have remnants اللي هم البقايا. كل هذول molecules will be transported to the liver. هلا basically the glucose is going to be converted to glycogen لأنه في عندنا well fed state, so the glycogen synthesis is going to be active, as well as we will have a glycolysis process, and this is very important. وهذا الشيء غريب إنه يكون في عندك glycogen anabolism and catabolism, and we will understand why is that, as well as the hexose monophosphate pathway, another mechanism of the NADPH generation because the NADPH is very important for the anabolism. Okay. هلا الخلايا الأخرى إما بتعمل anabolism أو catabolism فهي أمام خيارين بينما الليفر has these two options. Okay. The amino acids are going to be converted either to proteins اللي هذول البروتينز راح يكون جزء مهم بالمستقبل عشان نكون ال VLDL. Okay. Some of these amino acids are going to be metabolized to produce certain metabolites, بعض الهرمونز, بعض other forms of amino acids, and due to this we will release ammonia, and these ammonia molecules need to be detoxified by urease cycle, واحنا هون مش شايفينها بس هي ضمنيا موجودة. Okay. And some of the amino acids can be degraded to produce acetyl-CoA or pyruvate or certain components of the Krebs cycle due to the digestion. And eventually, the carbohydrate molecules and the amino acids will be converted to fats. Why is that? Because you have high concentration of acetyl-CoA. And why you are generating fats basically in the liver? This is because the major function of the liver, one of the major functions, is to produce fats to supply or to fuel the body. لازم يكون عندنا fats عشان نزود الجسم بالطاقة. So in order to do that, you are using all of these components to synthesize fats as well as storage of glycogen لأن أنت في حالة wolfid state. Okay. هلا هذول fat molecules راح يتصدروا على شكل VLDL molecules اللي هم very low density lipoproteins and then will be sent to the adipose tissue. In general, consumption of these molecules will cause obesity in the future by the help of the liver itself. لأن الليفر هو مش مسألة يعني مش وظيفته إنه يعمل فات، هو وظيفته إنه يعمل metabolism لهذول المولكيولز. So this is very important concept to remember. On the other hand, we have fasting state or starvation state, which means that you are going to consume your glycogen to produce glucose, as well as you have to do gluconeogenesis. So you have here catabolic pathway and anabolic pathway صارت بعكس تماما الحالة الأولى اللي شفناها. Okay, but still the liver can do anabolism and catabolism at the same way in order to produce a glucose molecule for other tissues. عشان هيك هو بحافظ على الأذر بيعمل supply للأذر tissues with the glucose. آخر شيء بدي أحكي عنه اليوم عشان نخلص موضوع نكمل المرة الجاية اللي هو the pyruvate molecule that is going to be converted from your amino acids basically اللي موجودين في السوليوشن نتيجة digestion of lipids اللي في البادي. وواحد راح يقول لي طيب ما احنا اصلا في حاله starvation. During the starvation you start to break down your lipids and you will generate these molecules هم الجليسرول واللاكتيت, and these molecules are going to convert it to pyruvate and acetyl-CoA, and eventually you will generate fatty acids, ketone bodies. And the ketone bodies during starvation or fasting are very important. هلا كيف صار في عندي فاستنج وتكسير للدهون؟ الدهون اللي في الجسم يا اخوان مش الدهون اللي جاي من الدايت. Okay, so all of these components are going to supply the production of either glucose or ketone bodies. ليش أنا بهمني هدول التو molecules إنه الليفر يصنعهم في حالة الفاستنج? Because these molecules are very important for certain types of tissues, specifically nerve cells. They rely on, they can use glucose as well as ketone bodies. لما يكون عندي fasting وبيقل لي جلوكوز بعد فترة من الفاستنج أو starvation, we rely on the ketone bodies اللي هم راح تتكون نتيجة الأسيتايل-CoA accumulation due to the fat digestion. اليوم بنوقف لأنه لازم أطلع محاضرة حتى المحاضرة اللي بعدية يدخلوا بدون ما مشاكل بالبلوك. Thank you for listening. ونشوفكم إن شاء الله بتاريخ 2/11 في عندنا محاضرتين إن شاء الله.